Okay.
The organelle we decided to present on was the Golgi Apparatus by Matthew Butler and Kathy Birch.
Okay, so the location of the Golgi apparatus is it's found in the cytoplasm of the cell, and it is actually right next to the endoplasmic reticulum. So they're basically right next to each other because they share a job together. And then it's also found near the nucleus, which is in the center of the cell. So they're like right next, like they're neighbors. They're really close to each other. And so the reason why it's there is because part of its job is to make protein with the endoplasmic reticulum. And so it actually transports into the Golgi apparatus from the endoplasmic reticulum. So like the location is very important because if it wasn't next to the endoplasmic reticulum, it wouldn't be able to perform its job properly. And if it was found elsewhere in the cell, it wouldn't be able to like function properly either.
Can you point it
As
out?
you have this diagram, it's right here. It's this orange piece in the middle.
Okay, so the function of the cell is it has one main job. It modifies, sorts, and it also packages the lipids or proteins for secretion. The secretion is when basically a cell substance is discharged or it's released from the cell. And so what happens is that from the endoplasmic reticulum, as I said earlier, since it's right next to it, it basically releases the lipid from the endoplasmic reticulum. So once the lipid leaves the endoplasmic reticulum, goes into the Golgi apparatus, and then it's absorbed, and then it later basically discharges or releases it, and that's basically the whole process of secretion. So it leaves the cell, and what happens once the lipids leave the endoplasmic reticulum is once it enters the Golgi apparatus, it turns into a secretion vesicle. So then once it turns into a secretion vesicle, which is basically the substance that it creates, like the cell substance or secretion that is created in the Golgi apparatus, it's then released again.
So it's pretty sequential then, right? You can see the arrows there that say this happens first, then this, then this.
Yeah. One of the one group of things that are like modified in the Golgi apparatus and then um, secreted out are hormones such as testosterone and estrogen. And the Golgi apparatus can even modify the phospholipids in the cell membrane if they need to be, like if they're down. Who was it discovered by? The Golgi apparatus was discovered in 1898 by Camillo Golgi. He first named it the internal reticular apparatus, and it was later named the Golgi apparatus because of who discovered it. It was discovered while he was performing an experiment with nerve cells, and he found it with an electron microscope. And what is it made up of? The Golgi apparatus is made up of flattened stacked pouches called cisternae. These are the things in which the proteins are modified and then pushed to the vesicles. And the vesicles are what carry the proteins from the endoplasmic reticulum to the Golgi apparatus and then out. So it comes in as like a protein vesicle, and then it'll leave as like a secretion vesicle.